When I previously depicted the banana cropping system, I showed a bare soil. The work we have been doing in Australia has been looking at the role of non-banana plants in the management of the disease, and they can be either deleterious, promote the disease, or beneficial in suppressing the disease. In our production systems, we want to reduce the FOC inoculum levels in the soil. We also want to increase the beneficial organisms that are antagonistic to the pathogen. Therefore, we need to understand the role non-banana plants have as alternative hosts which may carry infection through to the next crops. We also need to know what we can grow as rotation crops to get the greatest reduction in FOC inoculum and foster those beneficial organisms. And following planting, we need to know what we can grow as vegetative ground covers to stimulate and encourage beneficial organisms to keep a continuously returning production system going for as long as possible. To determine the alternative host status of Fusarium on bananas, we conducted multi-location field surveys of infected banana fields looking for different races of the pathogen. The surveys were conducted in 3 different production regions with distinct geographical characteristics. The frequently identified plants from the field surveys were included in inoculated glasshouse experiments which tested over 50 different potential alternative host species. From the glasshouse experiments conducted, all races of FOC could be recovered from the different plants, but some plants were found to be better hosts than others. Jay Anderson, who is part of Liz Aitken's team at the University of Queensland, additionally investigated colonisation of alternative hosts using a GFP-modified subtropical race 4. Jay found limited colonisation of the roots by the fungus, finding occasional hyphal nets, chlamydospores, and some colonisation of epidermal cells. However, when Jay treated the plants with herbicides, the GFP Fusarium was able to extensively colonise the dead weeds, with mass sporulation occurring. From the work that has been done on alternative hosts, a grower's guide has been produced. The guide gives some basic information like name, location, description, and a rating based on glasshouse results and whether it has been isolated from the field or not. The aim is to give growers some idea of the potential host status of the different weeds and whether they are likely to carry the disease over to the next banana crop. We can also consider non-banana plants as rotation crops to reduce inoculum and increase antagonistic organisms. In a glasshouse experiment using a method developed by Wayne O'Neill, we call pots in pots experiment, which is depicted in the figure across the top, we tested a range of potential rotation crops. The recovery of subtropical race 4 was compared in relation to our inoculated and uninoculated banana controls and was giving a range of responses. Some crops, like peanuts and brassicas, appear to be good hosts to FOC. There are many intermediate hosts, but we found 4 species that had a similar recovery of FOC to the uninoculated banana control, such as Dallas grass, Queensland blue couch, burgundy bean, and leucaena. These are worthy of further investigation. In a field experiment conducted by Charles Mintoff and his team in the Northern Territory, several potential rotation crops for the region were tested. I'm only presenting the results from 4 treatments: a weedy fallow, which was our control, cavalcade, Centrosema pascorum, jarrah-grass, Digitaria meligiana, and sugar graze, a forage sorghum. Charles used a method developed in a previous project to measure the FOC TR4 DNA based on a TACMAN assay. The samples corresponded to the previous crop of infected bananas. the rotation period when different crops were grown, and the subsequent banana crop following the rotation. And there was very little difference, and it certainly wasn't significant, in the TR4 DNA recovered from the soil between the different treatments and between the different phases in the crop cycle. However, there were differences in the disease incidence determined by the external expression of the disease in the subsequent banana crops. If we use the weedy fallow as our reference, there was a rapid increase in symptoms 21 weeks after planting, reaching around 40% at the harvest of the plant crop. Cavalcade was significantly slower to express the symptoms and had 20% expression by harvest. The jarrah grass was more rapid, reaching 50%, and the sugar graze intermediate. While the rotation crops didn't reduce the pathogen in the soil, as all treatments were equally infected, the cavalcade did slow the expression of the disease. From some earlier work which I published with Emily Rames, we found that when vegetative ground covers were maintained around the base of plants, there was a reduction in the amount of necrotic tissue within the banana pseudostem. Although the plants were still infected, necrosis was significantly reduced over 3 crop cycles. Associated with the reduction in pseudostem discoloration was an increase in fungal richness under the vegetated ground cover treatment over that same period. Fungal richness was also found to be associated with activity of the enzyme beta-glucosidase, so that an increase in fungal richness was highly correlated with increasing beta-glucosidase. And both an increase in beta-glucosidase and fungal richness were strongly correlated with a decrease in pseudostem discoloration using a 1 to 6 scale. Some preliminary results from a second field experiment conducted in North Queensland used Illumina next generation sequencing to track changes over a 4-year period in a field experiment. In this case, we used the number of Fusarium oxysporum OTUs as a surrogate to FOC because of the quarantine restrictions on working with Fusarium wilt in Queensland. Following establishment of the treatments, there was an increase in Fusarium oxysporums in the bare soil, the silver line, compared with the vegetated ground cover treatment, the yellow line. At around 36 months, the 3-year period after planting, there was a significant increase in the abundance of Fusarium oxysporum OTUs in the bare soil, which declined at around 42 months. At around the 31-month period after planting, there were very serious unfavourable climatic conditions. Firstly, heavy saturating rain, followed a few weeks later by extremely hot, dry conditions. And we will be investigating further the impact of those climatic extremes leading to the dominance of Fusarium oxysporums in the soil, but also the buffering effect created by ground cover.